Welcome to the Biology Brief channel. Today we are for MCQ discussion part 2 of the chapter Molecular Base of Inheritance. Today in this class we are going to discuss about 30 important MCQs of the topics transcription, genetic code and translation. Let us start. First question. Select the process that involves the polymerization of both ribonucleotides and deoxyribonucleotides. Listen, the polymerization of both ribonucleotides and deoxyribonucleotides. If you examine the options, you can easily arrive at the correct answer as DNA replication. During the DNA replication, RNA primer is formed by the polymerization of ribonucleotides and new DNA segments are also formed by the polymerization of deoxyribonucleotides. What about other options? During transcription, mRNA is formed by the polymerization of ribonucleotides. What about translation? During translation, polypeptide chain is synthesized by the polymerization of amino acid residues. What is RNA processing? RNA processing or RNA splicing. Here, the intronic sequences are cut and removed and the exonic sequences are joined. There is no polymerization during RNA processing. So, our answer to the first question is option 2, DNA replication. To next question. Given below are two statements. Statement number 1. The process of copying genetic information from one strand of the DNA into RNA is termed as transcription. Statement 1 is correct. It is a definition of transcription. Here, one strand of DNA is copied into mRNA. Correct. Statement 2. A transcription unit in DNA is defined primarily by the three regions in the DNA. There are three regions in the DNA for transcription unit. One is promoter, then structural gene and terminator sequence. Here RNA polymerase bind with the promoter sequence and the structural gene is transcribed, and when the enzyme reaches terminator sequence, the transcription stops. Here in the statement 2, the statement 2 is also a correct one. So we can answer this question as by the option 1. Both statement 1 and 2 are correct. Option 1 is our answer to this question. You can easily write the correct answer by careful examination of the statements. Moving to next question. Transcription is the function of RNA polymerase enzyme. It polymerizes RNA nucleotides by forming. You know that DNA, one of the segment of DNA is transcribed to form mRNA. By the process transcription, here the enzyme is RNA polymerase enzyme. Here there is polymerization of ribonucleotides. Ribonucleotides are polymerized. Here 3 prime, 5 prime, here 3 prime, 5 prime. Here when two ribonucleotides are polymerized, a bond is formed by connecting 3 prime and 5 prime ends incorporating phosphate group. Here the bond is phosphodiester bond. Phosphodiester bond is formed. The bond formed here is phosphodiester. So you can select option 3 or 4. Then when transcription takes place, the process obeys the complementarity rule. The transcription takes place according to complementarity rule. So our option to this question will be option 3, that is phosphodiester bond is formed following rule of complementarity during transcription by RNA polymerase enzyme. So option 3 will be answer to this question. Let us move to next question. Fourth one. Which statement is not applicable to template strand in polypeptide chain synthesis? Which statement is not applicable to template strand? Template. DNA template. It has 3', 5' polarity. The statement is a correct one. The template strand of DNA has 3' to 5' polarity. Option 1 is correct. It takes part in transcription. Template strand takes part in transcription. The other strand runs in 5', 3' direction. It is called a coding strand. This is template. Nucleotide sequence is complementary to one present in mRNA. Nucleotide sequence is complementary to one present in mRNA. Here the mRNA is formed. mRNA is formed in 5', 3' direction. mRNA. Here mRNA formation is according to the rule of complementarity. So nucleotide sequence is complementary. The third statement is also correct. It is also known as coding strand. Template strand is not called a coding strand. The other strand is called a coding strand because the nucleotide sequence is in mRNA similar to coding strand, not similar to template strand. It is complementary to template strand, similar to coding strand. So template strand is also called a non-coding strand. So the template strand is not known as coding strand. It is known as non-coding strand. So fourth option will be our answer because that statement is not applicable to template strand. Okay. Let us move to next question. Read the given statements and choose the correct option. If you examine the options, first two statements are contradictory. So only one will be correct. In transcription, only one strand of a segment of DNA is transcribed. In transcription, both the strands of a segment of DNA are transcribed. Complementary statements. Here, we know that during transcription, only one strand of DNA segment is transcribed and the transcribing strand is known as template strand. First statement is correct, second is wrong. Then third and fourth statements are also complementary, contradictory statements. Let us see. Once replication process sets in, both the strands of segment of DNA is replicated. Once replication process sets in, only one strand of segment of DNA is replicated. When replication process sets in, both the strands of segment is replicated is correct option, correct statement. Fourth one is wrong one. So we have to choose correct options. What is our answer? Statement 1 and 3. Statement 1 and 3 are correct. Fourth option will be our answer to this question. Let us see next question. Sixth one. Statement type question. In prokaryotes, the structural gene in transcription unit is mostly monocystronic. It's a wrong statement. In prokaryotes, usually the structural genes are polycystronic. The monocystronic structural genes are present in eukaryotes. The monocystronic structural gene has interrupted coding sequences called exons. Interrupted non-coding sequences called introns. As such, statement 2 is wrong. If you rewrite it, the monocystronic structural gene of eukaryotes has interrupted with the non-coding sequence called introns. The correct one. So both the statements are here wrong. So our answer to this question is option 2. Both statement 1 and statement 2 are wrong. Option 2. Let us move to next question, 8th one. In one strand of DNA has the base sequence ATCC, ACGA, CTAG, and the second strand undergo transcription. What would be the base sequence of mRNA. Here this is one strand. This is one strand. The second strand undergoes transcription. This strand undergoes transcription. So this will be template. This will be template, and this will be non-coding also. Non-coding also. Here this strand is coding strand. This is coding strand. New mRNA is formed. mRNA is formed. The train-based sequence in the mRNA, it is mRNA. mRNA will be same as the coding strand. There is one exception. In the case of thymine, there will be uracil. So you please note the position of thymine in the coding strand. You can write the nitrogen-based sequence of mRNA directly from coding strand as A. For U, for T there is U. C, C, A, C, G, A, C, U, A, G. A, U, C, C, A, C, G, A, C, U, A, G. This is the nitrogen-based sequence of mRNA. Examine the options AU, CAC, GAC, UAG. Third option is similar as this mRNA sequence. So our answer, you can find out easily. If you know the theory part, you can easily find out the answer, correct answer. Here third option is our answer. What about next question? It is also from transcription. Write the sequence of RNA transcribed from DNA which has the coding strand 5' ACC AGG TAA CAAG 5'. This is the coding strand. This is coding strand. What about non-coding strand? Non-coding strand will be in the direction 3' to 5'. This is non-coding. Non-coding strand and also template strand. Then mRNA will be formed, which is 5', 3' direction. And you can write the nitrogen-based sequence of mRNA. This is mRNA directly from the coding strand, as in the previous question. ACC, AGG, then check T, right here U, AAC, AAG. Then examine the options. You can strike out option 1 and option 3 because they are in the direction 3' to 5'. Here mRNA will be formed in 3' to 5' direction. So you can eliminate option 1 and 3. Then what about option A? ACC, AGG, UAA, CAG. Fourth option is, you can directly find out the answer and select the option for this question. Option 4 will be the answer. Move to next question. Which one is the correct product of DNA dependent RNA polymerase to the given template? Here template is given. This DNA template is given. Template in the direction 3' to 5'. So you have to write the coding strand. You can write the coding strand in 5', 3' direction. T complementary to 2 is coding strand is the DNA. T, A, AT, G, T, A, C, C, G, T, 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 A, T, A, G, G, T, A, G, T. This is the coding strand which is complementary to this DNA template. From this, you can arrive at the nitrogen-based sequence of your mRNA, mRNA. Only thing you have to do is replace thymine with the uracil. So A, U, G, U, A, C, C, sorry, G, G, C, U, 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 A, U, A, G, G, U, A, G, U. Here it is a lengthy mRNA. Then see, here all options are in the direction 5' to 3'. So AUG, AUG, you can strike out this option. AUG, UAG, AUG, UAG, AUG, AUG, CC, CCG, AUG, UAC, AUG, UAC, UAC, CGU, CGU, UUA, UUA, UAG, UAG, GUA, AGU. If you write correctly, carefully the answer, you can easily arrive at option 1 as correct answer. Option 1 is correct answer to this question. Here, these previous three questions are questions from transcription and different models. You can practice more questions for the exam. Next question, 10th question, statement type question. Read the following statements and choose the correct statements. RNA polymerase associates transiently with a rho factor to initiate transcription. The statement as such is wrong because RNA polymerase associates transiently with the sigma factor to initiate transcription. Statement 1 is wrong one. In bacteria, both transcription and translation takes place in the same compartment. The statement is correct because bacteria is a prokaryotic organism. There is no division into nucleus and cytoplasm. The cell is a prokaryotic one. So, transcription and translation are coupled and it takes place simultaneously in the cell in the cytoplasm. Option statement 3. RNA polymerase 1 is responsible for transcription of tRNA in eukaryotes. RNA, you know that in eukaryotes there are three types of RNA polymerases. There are three types of RNA polymerases. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. RNA polymerase 1 is responsible with the synthesis of ribosomal RNA. 2 is responsible for the synthesis of hnRNA or the precursor of mRNA, and polymerase 3 is mainly synthesize tRNA. So RNA polymerase 1 is synthesis of rRNA, not tRNA. So this is a wrong one. This is a wrong one. Then statement 4. When hnRNA undergoes capping process, adenylate residues are added at the 3' end in a template independent manner. hnR undergoes capping. Here, the RNA processing, during RNA processing, capping is formed. Capping is formed at the 5' end of hnRNA in a template independent manner. The statement is as such is wrong one. The precursor of mRNA is in eukaryotes is hnRNA. hnRNA is called primary RNA, which is the precursor of mRNA. There, the mature mRNA is called secondary RNA. So, statement 5 is correct. So, you have to choose correct statements. Here, statement 2 and 5 are correct. So, our answer is option 4. 2 and 5 only correct. Moving to next question. In eukaryotes, transcription is done by 3 classes of RNA polymerases. We already mentioned. Of these, the group of RNAs transcribed by RNA polymerase 3 is RNA polymerase 3. RNA polymerase 3 mainly transcribe tRNA in eukaryotes. Besides tRNA, the small nuclear RNA is transcribed and 5S rRNA. In eukaryotes, rRNAs are formed by polymerase 1, RNA polymerase 1. But rRNA, 5S rRNA is transcribed by RNA polymerase 3 along with the tRNA and small nuclear RNA. So, our answer is option 4. tRNA, 5S rRNA and snRNA, small nuclear RNA. These are transcribed by RNA polymerase 3. Our answer is option 4. What about next question? Which one of the following is not transcribed by RNA polymerase 3 in eukaryotes? We already saw the question. RNA polymerase 3 polymerases in eukaryotes, tRNA, small nuclear RNA and 5S rRNA. hnRNA is transcribed by RNA polymerase 2 in the case of eukaryotes. Our answer here, not transcribed. Our answer is fourth option. And what about 13th question? Analyze the statements and choose the incorrect one. Cistron is a segment of DNA that code for a polypeptide. Cistron is the smallest segment of DNA which can code for a polypeptide. Correct statement, which is also the definition of cistron. Exons are coding sequences present in hnRNA. Exons are coding sequences present in hnRNA. hnRNA is primary RNA present in eukaryotes. Exons are coding sequences. The statement is correct. Introns are intervening sequences. Correct up to this much. Present in mature RNA. In mature RNA, introns are not present. Before that, the introns are removed from hnRNA by RNA processing. So, introns are the intervening sequences which are non-coding sequences present not in the mature RNA, present in the hnRNA. The statement is a wrong one. Splicing represents the dominance of RNA world. We know that before the evolution of DNA, RNA dominated in the living world. For remembering the dominance of RNA, there are some processes in molecular genetics. For example, RNA splicing and the protein synthesizing machinery is also revolving around RNA. These are symbols of dominance of RNA world. The fourth option is also a correct one. Our answer to this question is the wrong statement, option 3. Next question. Which of the following is correct regarding RNA processing? Correct regarding RNA processing. Let us see option 1. In capping, methyl guanosine triphosphate is added to the 3' end of hnRNA. It is a wrong statement. Capping takes place in the 5' end of hnRNA. During capping, methyl guanosine triphosphate added to the 5' end of hnRNA. Option 2. RNA polymerase 1 transcribes tRNA in eukaryotes. It is also a wrong one. RNA polymerase 1 transcribes rRNA in eukaryotes and tRNA is transcribed by RNA polymerase 3. In tailing, adenylate residues are added to the 3' end of hnRNA. Correct statement. Poly A tail is formed at the 3' end of hnRNA during RNA processing. Correct statement. Then, 3 types of RNA polymerase catalyze the transcription of the 3 different types of RNA in most bacteria. The statement as such is a wrong one. 3 types of RNA polymerases are not present in prokaryotes. 3 types of RNA polymerase catalyzing the transcription of 3 different types of RNA is present in eukaryotes. The statement is wrong one. You have to select correct statement, our correct statement in this question is option 3. Our answer is option 3. Moving to 15th question, select the correct statement about mRNA from the following. mRNA, regarding mRNA, you have to select a correct statement. Eukaryotic mRNA is usually polycistronics. Wrong statement. Eukaryotic mRNA is usually monocistronic. What is polycistronic and monocistronic? If the structural gene code for many genes, many genes in a single structural gene, it is called polycistronic. In eukaryotes, a structural gene consists of a structural gene is capable of producing single polypeptide. So it is called a monocistronic. Monocistronic mRNA is more common in prokaryotes. Also wrong. Monocistronic mRNA is more common in eukaryotes. Eukaryotic mRNA has methylated region at 5' end. That is correct one. Eukaryotic mRNA means hnRNA. In eukaryotic hnRNA, methylated region, methylated guanosine forms head region at 5' end, and the statement is correct. It is the most abundant form constituting 70 to 80% of total RNA. mRNA is not most abundant form of RNA. Most abundant form of RNA in a cell is ribosomal RNA, which is up to 70 to 80%. The second abundant one is tRNA. mRNA is the least abundant form. So the statement 4 is also a wrong one. Our answer, we have to select the correct statement. Third one is the correct statement. Our answer is third option. Next question. The salient features of genetic code are. The question is about genetic code. The salient features, the unique features of genetic code are. First one, A. The code is palindromic. This is a wrong one. The code is triplet. The code is triplet, not palindromic. Wrong option. UGA acts as initiator codon. UGA. We know that UAG, UGA and UAA are stop codons. These are stop codons. UGA not act as initiator codon, it is a stop codon. That statement is also a wrong one. The code is unambiguous and specific. Correct. Genetic code is unambiguous. No ambiguity regarding the specification of genetic code. Genetic code is clear and it is specific also. It is a code specific also. Correct statement. D. The code is nearly universal. It is a statement as such in this textbook. Genetic code is nearly universal. It is applicable to majority of all living organisms except a few options. Option E, so it is also correct. C correct, D correct. What about option E? There are 64 codons. Correct. Up to this level, there are 64 codons, 4 amino acids. Totally, there are 64 codons, of which 61 code for amino acids. 3 do not code for amino acids. The codons which do not code for amino acids are called stop codons. Stop codons, UAG, UG and UAA. 64 amino acids, 64 codons code for amino acids. The 64 codons which code for amino acids are called sense codons. The stop codons which do not code for any amino acid are called nonsense codons. Sense of the 64 codon, 61 are sense codons, 3 are nonsense codons. So there are 61 codons for amino acids will be correct statement. This statement as such is a wrong one. You carefully analyze the statements and find out correct answer. Here for this question, The statement C and D only are correct. There is third option for misleading you. C, D and E only correct. Misleading option. Carefully answer. C and D are correct statements. Our answer is option 4 for this question. What about next question? Given below are two statements. One is assertion and the other is reason. Assertion. UAA, UAG and UG are called terminator codons. We saw earlier in the earlier question. These three are called stop codons or terminator codons because they terminate or they stop protein synthesis. Reason, terminate codons are recognized by protein releasing factor. When terminate codons come, the protein releasing factors recognize them and they help for the removal of polypeptide chain. So, reason is assertion and reason are correct. Reason is correct explanation of assertion. So, first option will be our answer for this question. Yes, let us move to next question. Which of the following is correct regarding genetic code? Again, question from genetic code. UUU is the initiation codon. Wrong one. We know that AUG is the initiation codon. UUU code for phenylalanine, but AUG code for methionine. AUG is the initiation codon and it codes for methionine. So, statement 1 is wrong one. There are 64 codons and also 20 amino acids. There are 64 codons and 20 amino acids. Correct one. Correct statement. For translation, the raw materials are 64 codons and 20 amino acids. Correct statement. Statement 3. 3 random nitrogen bases specify the placement of one amino acid. Random is a wrong term here, not random. 3 continuous nitrogen bases specify the placement of one amino acid. So, the statement as such is a wrong one. Fourth option, UAA is a nonsense codon. UAA is a nonsense codon or UAA is a stop codon. Up to this, the statement is correct. Read the remaining, which also codes for methionine. Nonsense codons do not code for amino acids. Here, code for methionine is AUG. Methionine has only one codon and that is AUG and not UAA. So that statement, fourth statement is also wrong one. If you analyze the four statements, you can see that option 2 will be correct and it is the answer to this question. Option 2 is our answer. Next question, 19th one. Statement 1. A non-overlapping code means that a base in a mRNA is not used for different codons. If this is an mRNA, 5 prime, 3 prime, mRNA, AUG. Here it is a non-overlapping one, AUG. Next one, GUG, for example, here. AUG, the first 3 nitrogen bases code for one amino acid, we know that, methionine. Here there is no overlapping. So this code or any nitrogen base of this codon cannot be used for different codon, another codon. So that is a peculiarity of non-overlapping code. Non-overlapping code will be specific for amino acids. Non-overlapping code means that the base in mRNA is not correct. Statement 1 is correct. What about statement 2? In translation of mRNA, the codons do not overlap. In translation, mRNA, the codons do not overlap and read subsequently. They read subsequently or continuously or constantly without any overlap. Statement 2 is also correct one. So, statement 1 and 2 are correct. So, answer to this question is option 1. Both statement 1 and statement 2 are correct. What about 20th question? It is a matching question and it is from genetic code. Genetic code. Here some codons are given. Some codons are given in column 1 and amino acids are given in column 2. GCA, UCA, UGG, GAG, UGF. Here GAG, you all know that GAG. When GAG is converted or mutated to GUG, GAG is for glutamine or glutamic acid. Here, when A, single base substitution, here valine will be replaced. When in insulin, when in hemoglobin, glutamine is replaced by valine, there is sickle cell anemia. You know that the fact very well. Here GAG code for glutamine. So D will be matched with 3. Here D3 is for option 1 and 3 only. 1 and 3. Here you can write the code number for the amino acids. Code number UCAG 1, 2, 3, 4. Code number for GCC code number 422. UCA code number 123. We know that the middle number is 2, so the column is second column. You can imagine the amino acids of second column. There is a code SER proceed through allothur. Serine, proline, threonine and alanine. Here 422. 4 means fourth row alanine. 422. GCC codes for alanine. We can easily decide from this code number. 1, 2, 3. 1 means first row, that is 1, 2, 3 will be serine. UCA will be serine. Then A, alanine, A2 is third option. A2, alanine 2, A2. What about B? B5, A2, B5, C1, D3, E1. Third option. You can easily find out the answer from this clue. You refer other class where I have already taught how to answer questions from genetic code very easily. You can refer that video also. Yes, you can find out answer very easily like this. I think you understood the answer. Moving to next question. 21st question, statement 1. In prokaryotes, the transcription and translation are coupled as they take place in the same compartment than the cytoplasm. There is no compartmentalized in the nucleus and the cytoplasm in the case of prokaryotes. So transcription and translation coupled and they takes place in the same compartment. Statement 1 is correct. Statement 2. RNA polymerase 2 transcribe rRNAs. In eukaryotes, RNA2 polymerase transcribes hnRNA or primary mRNA. So statement 2 is wrong one. We can answer statement 1 is correct, but statement 2 is wrong. That means option 3 will be our answer to this question. 22. The substitution of amino acid in the globin protein results due to a single base substitution of 6th codon of beta globin gene from GAG to GUG. Already we mentioned GAG. GAG is glutamine or glutamic acid. GUG is valine. Here the amino acids referred as respectively. Here glutamine, glutamic acid and valine. Option 2 will be our answer. You can easily find out this writing their index number also, code number also. Code number UCAG 1, 2, 3, 4. GAG is 434. third in the third column, fourth row. Valin GUG. GUG will be 4, 1, 4. That is first column, fourth row, first column, that is valin. This is glutamic acid. You can easily find out in both ways. Glutamic acid and valin is the answer. Second option is our answer. Next question. Given below are two statements. Statement 1. The first phase of translation is the activation of amino acids in the presence of ATP. And linked to their cognate tRNA is called amino acylation of tRNA. Very correct. The statement is very well correct. First step is amino acylation of tRNA or charging of tRNA. tRNA is charged using ATP and suitable amino acid is added and it goes to the rRNA for translation. Statement 2. There are about 20 different types of proteins are present in a ribosome. The sentence is copied from the textbook and the statement is also correct one. Statement 1 and 2 are correct statements. So our answer to this question is first option was statement 1 and 2 are correct. 24th question. Next question. It is also statement type question. Statement 1. The secondary structure of tRNA has been depicted as inverted L. Statement 2. In the actual structure of tRNA in a compact molecule which looks like say clover leaf. Here the descriptions are misplaced. The secondary structure of tRNA looks like a clover leaf. Here it is clover leaf. The actual structure or 3D structure is a compact molecule like inverted L. The description is misplaced. So due to this misplacement, both statement 1 and 2 becomes a wrong one. So our answer is option 2. Both statement 1 and statement 2 are wrong. Next question, 25th question. During translation of eukaryotes, the anticodon to be aligned with the initiation codon. Initiation codon, we know that initiation codon is AUG. Here AUG is in the mRNA and it is in 5' to 3' direction. The mRNA is scanned or copied by the ribosome in the direction 5' to 3' direction. This is the direction of reading of ribosome in this direction. Here the initiation codon AUG is coupled with the UAC, the anticodon UAC of tRNA. Here the UAC direction will be 3', 5'. So the anticodon to be aligned with the initiation codon is 3' UAC 5'. 3' UAC 5'. Second option is answer to this question. Very easy question. Carefully answer the question for your real examination. 26. Examine the following statements and select the incorrect one. First statement. A translation unit is the sequence in mRNA flanked by the start codon and stop codon. Correct statement. Translation unit consists of structural gene with a start codon and stop codon. Here the statement is correct one. An mRNA has some additional sequence that are not translated and are referred as untranslated regions or UTRs. Very correct. mRNA, the translation unit of mRNA also consists of some untranslated regions also. Statement is correct. The UTRs or untranslated regions are present only at 5' and it is a wrong statement. UTRs of the translation unit are present both in 5' and 3' ends. In both ends we can see the UTRs. That is the wrong one. The untranslated regions or UTRs are required for efficient translation process. The function of UTRs are making the translation process very efficient. So the statement is also a correct one. So we have to select incorrect from this option. So our answer is third option. Third option is our answer. Let us move to next question, 27th one. Find out the incorrect statement. You have to find out incorrect statement from the following. Amino acylation is essential for peptide bond formation. Peptide bond formation is an energy consuming process. Here, due to amino acylation, tRNA is charged using the energy of ATP. During amino acylation, tRNA is combined with its specific amino acid and used energy from ATP for the peptide bond formation. So option 1, statement 1 is correct one. After the process of peptide bond formation, the tRNA exits from the ribosome. When peptide bond is formed, the amino acid is incorporated and the free tRNA exit from the ribosome and go for charging again. Correct statement. The amino acylation is not a catalytic process. Amino acyl tRNA synthetase. For the amino acylation, it is a catalytic process, it is an enzymatic process. Here, the functional enzyme is amino acyl tRNA synthetase. So, statement 3 is a wrong one. Amino acylation needs ATP. We already said amino acylation energy is needed for charging of tRNA. Energy is needed. That energy is driven from ATP. So amino acylation needs ATP. It is also correct statement. Our answer is incorrect statement. That is option 3. Option 3 you have to select. Option 3 for correct answer for this question. Then 28th, match column 1 and column 2 correctly. You have to match. Then lysine. So let us start from tRNA. We all know that tRNA is the adapter molecule for protein synthesis. tRNA adapter molecule. Then mRNA after its function, it is degraded. So it is called a soluble RNA. mRNA, soluble RNA. Then lysine and arginine are basic amino acids which are the integral part of histone protein. Histone protein are basic proteins, positively charged proteins. They associate with negatively charged DNA for packaging. So lysine and arginine are basic amino acids. So you can match lysine with the option 3, basic. The remaining valine has the codon GU. GUU is the codon for valine. So our answer A3, B1, C4 and D2. Fourth option will be our answer to this question. 29th question. Given below is a sequence of nitrogen bases in mRNA of Escherichia coli. Identify the amino acid that would be incorporated at codon position 3 and 7 during the process of its translation respectively. You have to imagine the process of translation of this DNA. Here given mRNA of Escherichia coli is given here. When it undergoes translation, we have to find out the codon, the amino acids incorporated for the codons at position 3 and 7. You see, here the mRNA is written in the form 3' to 5' direction. Here, mRNA during translation read in 5', 3' direction. So you have to rewrite or reread it in reverse order. You have to read it in 5', 3' order. That means first codon from reading from 5' will be AUG. Second AGC. AUG. Third one again AUG. Then fourth GUA. Fourth. 1, 2, 3, 4. GUA. Fifth GUG. Sixth UUU. Seventh, GGA. Then CUA, UAG. If you rewrite this in this direction, you can see that first codon is initiation codon, last codon is termination codon. 2, 3, 4, 5, 6, 7, 8, 9 codons are there in the mRNA. So we have to label position 3 AUG first, position 3, and position 7 GGA. Called AUG, we all know that initiation codon, it codes for methionine. This GGA is code number 443. Using UCAG, 443 is the code number. Fourth column, fourth row, that is none other than glycine. GGA is glycine. So our answer is methionine and glycine. Third codon codes for methionine, and seventh codon code for glycine. Fourth option is our answer. You carefully analyze the facts and reach correct answer for this question. Here reverse order is given, 3', 5' direction is given. Here the reading of mRNA is in 5', 3' direction. You have to reverse the order and find out third position and seventh position before arriving at the amino acids coded. Yes, our last question, 13th question, our last question. In Escherichia coli, the mRNA structural gene contains 100 codons, starting with the AUG and UA as the last one. Here it is, this is the mRNA. First codon is AUG, last is UAA. First one is 100th one. 100 codons are there. First one is initiation codon, last one is termination codon. Here, what happens? If 5th and 15th codons are mutated to UAG and AUG respectively at the center, this is the 5th codon, this is 51. 5th is mutated to UAG, 51 AUG. Select the possible outcome of the translation. Outcome of translation is added. Asked. Outcome of translation is added. Here you see this is initiation codon and this 15th codon is termination codon. When here the translation starts, here translation ends. Then 51 also initiation codon, UA is the old termination codon. So there will be 2 polypeptide chains will be formed. 2 polypeptide chains. So you can strike out option 1 and 2. 2 polypeptide chains are formed. What about the number of amino acids? 15th one is termination codon. No amino acid is added. So here 49 amino acids are incorporated. Here also UAA, 100th one is also a termination codon. There also no amino acid will be incorporated. There also 49 amino acids will be incorporated in the polypeptide chain. So what will be the outcome of translation? 2 polypeptide chains are formed with 49 amino acids each. Fourth option will be our answer to this question. So we have discussed some important questions from the chapter. Hope it will be very useful to you. Thank you very much.